Now, Marty, I apologize because I'm going to have to read your bio. But the title for today's meeting is Recent Findings on Ecology and Ectomycorrhizal Fungi Across Coastal BC. And Marty is a local on the island and is a West Coast regional soil scientist with the BC Ministry of Forests and is located in Victoria. Oh. Marty's areas of interest are soil ecology, especially ectomycorrhizal fungi, biodiversity conservation, carbon sequestration, and forest nutrition productivity. Marty is a member of the Provincial Soil Science Group and undertaking North American-wide studies on compaction and site organic matter removal, the long-term soil productivity study. Very interesting. Most recently, he has also been examining nitrogen and phosphorus deficiency inherent to coastal forests and their interactions on conifer nutrition and forest productivity. Amazing. Well, let's welcome Marty.
Yes, well, thanks for inviting me. It's nice to be back. I've done a few of these SPIM talks, and it's always wonderful to be in front of such enthusiasts for mushrooms. Since we've got lots of time, I've thrown in some pictures to start, and then unfortunately, I have to show you some graphs because as a scientist, that's the only way I know how to communicate is graphs. So I'll try and explain things as best I can. If it's really making no sense, it's maybe give me one of those looks and I'll stop. And Andy was doing that when I got out of the car, so I know that I'll rely on you to slow me down if it's not going well. So yeah, I've been with the Forest Service for 30 years now, and really got into fungi and mushrooms and symbioses, about 20 years ago. In the good old days, we would find these kind of mushrooms like this spring romaria. Magnapes is the name I tentatively put to it. And then we would try and key it out in a book, right? And then you'd not really know if you were right or not. And then maybe you'd dry it and send it to UBC to put in the herbarium, or maybe you would just throw it away. But it was pretty frustrating, right? You just never really knew if you had the right name for anything. But then came along the molecular revolution. And so for the last, I'd say, 12 years, I've been collecting, trying to get a nice photo, trying to find nice, fruitings like this, collecting photo, get a molecular sequence, which is essentially like a barcode. You've probably heard of that barcode of life kind of thing. And with that molecular data, you can now compare your mushroom with everybody else's mushroom. And so there's these nice public databases where you can throw it in and they'll show you all the collections that match yours. And there's one website that makes a map for you too. So it'll say, yes, this mushroom has been collected all over the place, right? So in this example, Magnapes is kind of interesting because I blast that. That's what we call it, right? When you sequence that, blast it. And it might match one or two other collections. So there's not much of it being collected over the years. And somebody else maybe have called it Magnapes. So I think to myself, okay, I guess I'm right. It is Magnapes. But then you go into the search and just out of curiosity, I ask myself, how many other collections are there that go by the name Magnapes? And there's like 5, right? So we're finding very similar mushrooms that everyone's keying out to this name, but when you compare the genetics, they're clearly distinct. So that's what we call cryptic species. And Romaria is full of these cryptic species. It's just all the same name keeps showing up in different collections. So clearly they're closely related, but they're distinct. And then another example, sort of on the other end of the spectrum, is Phlegmaceum glaucopus. So that used to be the Cortinarius group. Now they've split it in a bunch of different genera. It's basically a full-time job to keep up with all the taxonomy, right? And so glaucopus, when you sequence that and send it around, you'll find collections from all over the Northern Hemisphere. It's all over Europe and Asia, North America. So it's a widespread fungus, but at the same time, you don't come across it very often. So I might see it once every fall when I'm wandering around, once every 2 years, maybe. It's just not ever really, really abundant in my experience. So a lot of our fungi are like that. They're widely distributed, but always uncommon, which as an ecologist makes me wonder what's going on. What role can a fungus play when it's all over the place and never common, right? So we're in Victoria, and one thing I've been spending a lot of time doing too, is just trying to get a sense of how special is our local sort of fungal flora, right? And we're in kind of a neat area. This map on the right is kind of busy because it's got all these symbols, but this is kind of an ecosystem map that the Forest Service has pulled up. And I got this one because it extends south of us. So the yellow here is what we call the coastal Douglas fir zone, the really dry climate we have here in Victoria and a bit of the Gulf Islands. And my perception was always that this dry stuff just kept going all the way down to California. But in fact, there's quite a bit of moisture stands, not a huge amount of moisture, wetter stands, but anyways, greener stuff. You really don't hit more CDF till you get to the Willamette Valley in Oregon.
What are you talking about?
As a biome, this kind of low elevation coastal ecosystem, very, very dry, we go 12 weeks without rain. It's kind of unique. And I'm thinking in particular of this kind of upland sites, where we get Douglas fir, Salal, Oregon grape. And so, how many of our fungal species, and I'm obsessed with ectomycorrhizal fungi, but you could ask the same thing for saplings. How many of them are really quite unique to this type of ecosystem? And one way to kind of get at that is to sort of do that same thing. You find a mushroom, you blast it, and see where else it's been collected. So I'll just show you a few examples. Oh, I'm going the wrong way with this, sorry. So last spring, I collected this in Victoria, and I don't often see spring russula, so I thought that was kind of cool. It's not a particularly dense with features, this russula. It was kind of a pale white. I almost thought it was Russula cascadensis when I first saw it. But it was a nice spring one, so I just collected it, and I'll often get spore measurements and a few things like that and jot it down for laughs, but I had trouble keying it out. And I got the sequence and I sent it to Danny Miller. You guys might know Danny Miller. He's kind of my go-to for molecular. And it matched, there was nothing in the database that matched this. So right away I thought, okay, Marty Kranabetter, here I go again. but Daddy says, no, they have a couple collections that Ben Wu had, but they didn't post, they just got a short segment of the ITS, or this barcode. But it's not posted anywhere, but anyways, it matched that. So he calls it Wu 65, but this is a severely undersampled taxa, clearly. If I'm the only one who's posted it in a public database, and Danny's got a couple of. So that's one where I think, okay, maybe this russula is only found in these really dry ecosystems. And so that's kind of cool. And this is one of these gray tricholomas that are so challenging because, again, they don't have a lot. There's no veil or pungent odor or just something to really get there. I know exactly what this is. There's these gray tricholomas. I found this in Victoria as well. Sequenced it. There was maybe 3 other collections that matched it. We sort of used this European name, Scelteratrum, but it's clearly not the European one because when you look at the database, the European ones are way over there. It's genetically distinct. So this is a West Coast tricholoma that we don't have a name for yet. And I checked with Steve Trudell, who was here recently, I think, talking about tricholomas. And I said, what should I call this? And he says, I have no idea. So it's another one of these ones that I think might be only in these really dry ecosystems. And this is another spring Romaria. And again, I kind of keyed it out to Ruby Carnata. I really like these spring Romarias. It's a nice way to break out of winter, right? And go find these beautiful yellow Romarias. And it's the same as that Magnifies. Yeah, it's been collected twice before in sequence. And I should be careful when I say that because I'm sure it's sitting in herbariums all over the Pacific Northwest, but nobody sequences, right? So when I say there's only 2 other collections, that just means 2 collections that have been sequenced and posted. So, a couple more. And somebody called it Ruby Carnata, and I called it Ruby Carnata, and then I checked how many other species go by that name, and there's a lot. So once again, we're in this whole cryptic group where I don't know what we call it, but it seems to be in that kind of general area. And I think, again, this dry sort of CDF kind of fungus. And this is kind of a nice, again, it's from a Cortinaris, but we call it Phlegmaceum now, Citrinifolium. Has anyone come across this in their travels? I found it twice now in my life in Victoria. I think this was a fall one, yeah. It's got, when you cut it open, it's got quite nice pale flesh, like a pale lemon flesh. And kind of understated, but pretty at the same time. I think there might have been one other collection from California. Yeah, Okay, yeah. So it's one of those ones again where you think, okay, so there's this Cortinaria species that's only been collected a couple times. It's locally, we've seen it maybe 3 or 4 times in Victoria. What is that? How does that work? How has it got such a specific habitat? You know, they only find it in these areas, and what's it doing while it's here? Yeah, yeah. I've seen it. I've seen maybe 4 or 5 at one spot, but I wouldn't say dense. And but like I said, I've only collected it twice. This one, so this one was a little bit outside of Victoria in the Souk Reservoir, and we just parked the truck and I was collecting mushrooms for a UVic class, and lo and behold, this thing was fruiting away. It had this beautiful lilac edge. Can you see that? And a nice, really strong rim bulb. And I ran back and I tried to key that sucker out, usually mostly the European books. I couldn't put a name to it. And then I sequenced it and I sent it to the Cortinaria experts in Seattle and stuff. New species, yet again. So every year I find 2 or 3 new species. Like they just keep coming up, right? And of course, in the old, like I was saying earlier, in the old days, you would have put a name to that, either thrown it away or given it a UBC. You have no idea that this was never described or sequenced before. So that's almost the best part of my job, is just coming across these wickedly cool mushrooms that no one has documented before. And this is another one. It's a phelodon, which is teeth on the underside, right? And this was Andy and I were on a project a few years ago in the reservoir. We're looking at old growth stands and what was the total diversity of these things. And we came across this phelodon on root tips because we collected and sequenced ectomycorrhizal root tips. And it didn't match. I think it matched one collection from Pinus muriacata stand in California. So again, ridiculously undersampled. Oh, that's cool. So I went back to see if I could find a mushroom, and there were gobs of them. They were all over the place, this phelodon. So I collected these and sequenced it and confirmed it's definitely the one that's only been collected once ever in the history of mankind. And I'm just thinking, well, it's incredibly rare, presumably. Not that we've checked everywhere, but presumably we don't stumble across this thing very often. And then I'm finding all of them, right? It's like seeing a Sasquatch, and then you look and you say, well, no, there's actually 100 Sasquatches. It's a Sasquatch village, right? So it was so cool to just come across this. Again, nobody's named it yet. I have a long list of things that need names, but as a member of the Forest Service, I feel like it's just part of my job to document what is in our forests, right? We can't just assume somebody else is going to do it. And I like doing it. And then the other side of this, sort of looking for rare species, in the coastal Douglas fir zone, we do have these nice, rich soils here and there. So this is at Royal Roads. There's that, I don't know if you ever wander on that top plateau, there's that really silty, beautiful, lots of grand fir and cedar. So I like looking at these kind of less than common habitats, because the soil fertility will completely change the community of fungi in my experience. You're much more likely to find cool stuff on these sort of unique habitats. And the only other place I'm aware of that's close to this is at Mount Doug. There's some richer stuff near the ocean. So they're not common to come across these rich ecosystems. And so an ossibee, God help me, every anosbie I collect doesn't have a name. And this again was a new species. I just collected this last fall, and no record anywhere. Like I say, at least accessioned and put in these public databases. So that's another one that somebody's got a name. And a lot of the species, in my experience, in the West Coast, that we try and put sort of a European name to it, they're always, when you look at the distribution, they're always only from the West Coast. So they need new names. It's the whole anosbie group on the West Coast. If anybody here is thinking about grad work, I could get you into the anosbie. You like microscopes and chromatographs, you could do a nice bit of work on anosbies of the West Coast. And then this was another fun, beautiful Cortinaria, this nice cap and interesting shape. I think, again, there was no records of this in the database, but then one court expert told me the name. So they just named this, or just published this name recently. And I think there's 1 or 2 collections that they're posting. So I would assume this is one that's only found in these really rich soils, since I've never seen it anywhere else. Maybe it's with grand fir, but just another fun species that is just completely unknown. So really, and this was interesting. This little russula, and the russulas are so hard to deal with, right? They're always yellow spored and mild and purpley green cap, and this one's kind of on the small side. It's just a nightmare. So these molecular tools have completely helped us try and sort through russula. So I sequenced and blasted that, and it kind of matched this European species olivina. So I sent it to Danny, and he agreed that's probably where it is, but the match isn't that great, because normally with russula, it's at least 99% or better match. It's, they're really consistent that way. This one was like 98.8, I think, which makes me think that it's actually a West Coast species that we haven't described. I could be wrong, but if it was like 99.5 or better, then I would say, yeah, that's definitely the same thing. And I'd have to double check, but I don't think this has been recorded from, is that true, North America? Anyways, it's kind of a nice finding. And again, this was on a rich soil, maybe with grand fir, so it's just something we don't look at very often, right? And then the Ramarias, I'm convinced that grand fir is the ultimate host for Ramarias. And whenever I wander around a grand fir, I seem to find Ramaria. And this is the kind of reasonably common botrytis type. They're quite striking with that red colors and everything. And they can form a huge shiro. You can find dozens of these fruiting around these grand firs. And we found a few of these in the reservoir and stuff. So again, they're pretty uncommon, but not totally unknown. But when you sequence it, they're all over the place. There's a European botrytis, definitely not the one we have here. In the West Coast, we've got about 5 or 6 that all go by the name botrytis. So just another example of the cryptic species in Ramaria. We kind of give it one name, but there's probably half dozen unique species that look pretty similar. So again, if you're doing grad work after you're done the anosogby, then I would point you to the Ramaria to sort that out for us with the names of these. And I'm sure you all think you know what this is. Any guesses? Amphibious subroseus. Doesn't it look like subroseus to you guys? That's what I thought it was. But I collected this in Campbell River up in these montane stands I was working at. And I almost walked past it and I thought, wait a minute, there's no Douglas fir here. It's all amabilis fir and hemlock. And subroseus, I'm pretty sure, is only on Douglas fir. So I collect it. New species. It's only got one other match in the world from somewhere in Washington State. I sent it to Danny again, and yeah, nobody's described this stuff. So it's just fun, constantly fun. And so that's a high elevation Gymbidius. There's another one out there because I have these spore traps I'll get into later. And so I have 2 species of Gymbidius at that forest, neither of which have been properly named or described. So more work to be done. Okay, so that's my what's new in taxonomy part of the talk. So ectomycorrhizal fungi, that's my thing. Just to make sure we're all on the same page, these are the symbiotic fungi that you find on root tips on our major conifer species like pine, firs, Douglas fir, some broadleaves like birch and alder. And that's kind of what it looks like when you put it under the microscope. It's really this fungal mantle. I often think of it like a big sock that you put over your arm, right? It's just this thick mantle that covers the root tips. They have all different kinds of colors and shapes, and some have all kinds of emanating hyphae, and some are smooth, so they're all over the map. And every root I've ever looked at in the natural forest has had ectomycorrhizal fungi. There's sometimes in the literature people talk about non-mycorrhizal roots. I don't know what they're talking about. The only time you find a non-mycorrhizal root if you're growing a seedling in a nursery with all kinds of fertilizer and water, But in the real world, they're always mycorrhizal. And so it kind of raises the question, what do all these fungi do? And part of me asks this question because I'm just curious, but the other part of me thinks it's due diligence. As a member of the Forest Service, You know how we resource professionals talk about how we understand everything and we're sustainably managing things. The public should just calm down. It's all good. We're the professionals. We'll take care of it. And then you say, okay, you know everything. What is this fungus I see everywhere? What does it do? Well, I have no idea, right? And so I don't think we can claim to understand anything if we don't know what the symbiotic fungi of the forest, the very underpinning of the ecosystem, what do all these fungi do? And I don't know if that question is even answerable ultimately, but I just think if we're supposedly managing the landscape, the land base, we should have a better sense of what this guild does. So I'm going to show you a few examples of the studies I've been doing in the last 5 years or so. It kind of gets into this question, what are they doing? And why are they useful? So to start, historically, when you think about saprotrophic fungi like these white rot fungi on logs, they're quite a specialized group because they produce peroxidases. That's an enzyme that can break down lignin and cellulose and all these aromatic carbons. And so the thinking was these are the ultimate, decay fungi. They can break down anything. But historically, when you look at evolution, the thinking was when these saprotrophic ancestors became symbiotic, they lost some of that capacity. You can't be the ultimate destroyer of organic matter and a symbiote at the same time. That was kind of the thinking. So, but there's been some pushback on that. Some people are saying, actually, I think there is some evidence these peroxidases can be made by ectomycorrhizal fungi. Well, as it turns out, we have an experiment just outside of town in the Soup Reservoir that was almost designed to answer this question. It's called EP1130. And so it's a mixed species trial where we have Douglas fir and Western red cedar planted singly and mixed together, but properly replicated and randomly assigned to these treatments. And then the area where they did this study is about 12 hectares in size. And kind of a weird, lucky break for me is that it encompasses quite a wide range in soil fertility. So at this end, this is the low fertility plot. You can see this quite a bit skinnier Douglas fir, and the Western red cedar really quite scrawny. And as opposed to the high fertility soils, so way more nitrogen here, way less nitrogen. And the way we often capture that, my first graph, is carbon to nitrogen ratio. So the thinking is basically, if you have a lot of, imagine you're composting your garden with manure, you're bringing that CN ratio down. You're making it really, really fertile. And when you have a low CN ratio, you have a very high rate of nitrogen turnover. So the bacteria and fungi are just breaking that stuff down and releasing nitrogen into the soil, and the tree just takes it, no problem. But at the same time, if you start mixing straw in your garden, it's got a very high carbon to nitrogen ratio. It's starting to mobilize nitrogen, right? Things are trying to break it down, but there's a lot of carbon. So the release of that nitrogen comes down to almost zero, right? It's not, and your plants start to look a little sick. Nobody's happy. And what we then talk about is, well, at this lower end, you have nitrogen bound right into that straw in this example. And so the thinking is fungi will then just go into that organic matter, like basically colonize that straw directly and take the nitrogen right out of it. Won't wait for some other bacteria or fungi, saprotrophic fungi, to release it into the soil. It's just going to take it for itself, right? This direct uptake of organic nitrogen, which tends to happen when you don't have much nitrogen available to begin with. So this study, we have the two mycorrhizal guilds, ectomycorrhizal fungi on the Douglas fir, and then Western red cedar is an arbuscular mycorrhiza. It's a different kind of mycorrhiza that we don't think has these peroxidase abilities, okay? That's the theory. And so here's peroxidase on this axis. So that's that enzyme that's the destroyer of all organic matter. And this is my fertility gradient. I'm going from really high productivity soils to low productivity soils. And this is one tree species, and this is the other tree species. So you can see one tree species is pumping out all kinds of peroxidase, and one is not. Anyone care to guess which is which? You are right. So this is I haven't published this yet, but this is a really quite an elegant demonstration of this concept that Douglas fir, through its symbiotic fungi, can release nitrogen from this organic matter if the nitrogen is really bound tightly into the soil. And so it's actively mobilizing nitrogen, and I put where needed in bold, because I think one concept that keeps coming up that I rally against is that people think, oh, fungi do this or they do that, as if it's only one or the other. Yes, no. They make peroxidase or they don't make peroxidase. But clearly, on these productive sites, these are still fully ectomycorrhizal trees. There's all kinds of russulas and tomentellas and stuff down here. But why would a fungi make this expensive enzyme if it doesn't need to, when the nitrogen's already there? Whereas you get up into this neck of the woods and you have these pilodermas and other types of fungi. So there's quite a change in the community of fungi, and they're producing all the enzymes. So really, it's elegant. Mother Nature has been at this for a while, and the community aligns with that fertility gradient, and the fungal traits you need for that tree to thrive in that environment also aligns with that soil. It's not just a random, let's just throw an enzyme and hope for the best. And so why, you would think, if a little old guy from the Forest Service can just demonstrate that no problem, why has this been kind of a mystery for so long? Well, I think there's a couple things here. One is, so this is the piloderma. This is the one that I think is making a lot of that enzyme. Remember that this is a symbiotic fungi, so it's getting carbon from the tree. And I made this a big yellow arrow. So the tree has all kinds of photosynthate to send to its symbiotic fungi, right? There is no limit, basically, in the amount of carbon available for that fungi to do whatever that fungi needs to do. Whereas a saprotrophic fungi, like this Mycena, it's getting its carbon from the organic matter itself. So it's actually energetically very constrained, right? So I made this a smaller arrow. It doesn't have this tree sending it all the carbon it needs. It has to get carbon out of the soil all by itself. So it doesn't have the energy sources to kind of do what ectomycorrhizal do. And then the other thing we generally find is there's a lot of biomass in the soil that's, again, like ectomycorrhizal fungi. So even if the saprotrophs, pound for pound, were 5 times more capable of producing, say, this peroxidase, it doesn't matter to them because the saprotrophs are 1/10 the biomass. So even a inefficient fungus with this much biomass can still, make that soil do whatever it wants to do. So yeah, that combination of all this carbon from the tree and all this biomass, I think, is why we see such a difference between fir and cedar. What about phosphorus? Well, again, we're very fortunate here on the island because we have these rain shadow. So here in Victoria, we have very dry forests, but over here, it's very wet, right? Around Tofino, and I've got this map, wet maritime, moist maritime, dry maritime. So in a very short distance, like less than 100 kilometers, you can go from very dry to very wet. But 1,000 millimeters of rain a year to over 3,000 millimeters of rain a year. And the consequence is we get dramatically different soils. So on the dry side of the island, we have what we call brunissols. They're not particularly weathered because they're not getting the same amount of rain. But over in these podzels, you get this really spectacular orange horizon, this BF, BHF. And these podzels are chemically quite altered from all that rain over the last millennia. And so when we look at phosphorus availability, and this is Bray P, which is just like a typical agricultural thing you would do in your garden. In the brunissols, you tend to get something around 75 parts per million. There's lots of inorganic, fully available phosphorus that a tree might want in those brunisol soils. So around here, we generally don't think about phosphorus deficiencies for trees. Pretty good. Whereas on the podzols, barely get a heartbeat. There's almost no immediately available phosphorus in those podzolized soils. Very, very low. And so it's a little bit similar to the nitrogen story where we shift from inorganic P, all this inorganic P that's available, now to go to the organic P because the inorganic P is gone, but there's a lot of organic matter in those soils, lots of phosphorus in the organic matter. So much like nitrogen now, the fungi has to go get that phosphorus out of organic matter. And how does it do it? Enzymes again. And so these are big names, but that's a really important phosphorus removing enzyme, and that's another really important phosphorus removing enzyme. And lactase is another one of these, a little bit like peroxidase. It breaks down cell walls and things. And we sampled root tips in Douglas fir stands from the dry to the wet side of the island just to see what the species were doing. And, when you look at these brunosols, you're just seeing sort of a little bit of everything, but not a lot. And then you switch over these podzels and you start seeing the blue doesn't look as much because it's less to begin with, but the blue was at least doubled, twice as much of that enzyme. The monoesterase again about double, and the lactase really went up too. So when you get in these podzels, you start getting these like this Russula breviipes, this Lactaria subvisitus, Lactaria substratus. You start getting these species that are producing all kinds of these phosphorus acquiring enzymes. And so we tried, this is Justin Meads, who was a grad student on this and published this one. We found, this is the two enzymes again, that phosphorus 1, phosphorus 2. And what we found was a nice way of expressing this is the foliar nitrogen phosphorus ratio of the tree. So what that means is when an NP is really low, the tree has all the phosphorus it needs and it's demanding nitrogen. But when an NP gets high, it's much more stress for phosphorus. So down here, don't need phosphorus, not producing phosphorus enzymes, right? You get up here, starve for phosphorus, producing way more of these enzymes, both of them. And so very similarly to what I said about nitrogen, they're actively mobilizing P from soil organic matter where needed. And so again, it's these pods will select for these species. You don't find those species in the brunosols or vice versa. They're very separated, the communities. And those communities have these traits that reflect the constraining nutrient of those ecosystems for which a tree to grow. So it's just a slick system that these symbiotic fungi are constantly doing the lifting. So those two examples where there's nitrogen or phosphorus are very limited. Well, what about some of these really productive ecosystems where nutrients are very abundant? What's the story there? So this is a study I did at UVic. We had this thing called a MIFE, which measures nitrogen uptake on roots by ammonium and nitrate, so those inorganic forms of nitrogen. Long story short, we found on these richer soils, we found some of these anosebes, russulas, really high capacity for taking up ammonium, like the inorganic nitrogen in the soil. Whereas, interestingly, non-mycorrhizal roots barely work. So as I was saying earlier, people still talk about non-mycorrhizal roots as if it still kills me. In 2024 that we're in, this idea that why would a tree have mycorrhizae? There's already lots of nitrogen. It's just a waste of resources. But they're there because they're superbly adapted to pulling that nitrogen in. These rich ecosystems are very competitive, right? You log that site and you've got willow up over your head and shrubs and forbs and, salmonberry, like everything can grow in a rich soil. A tree has to be able to compete against all this other vegetation. And if you're a non-mycorrhizal tree, you won't last 5 minutes in that ecosystem. You need these fungi to persist and thrive. Now this graph, I brought this up because we're a mushroom crowd, so you might enjoy this, but it's a little bit complicated. This is looking at N15, which is a naturally occurring nitrogen isotope. So I didn't go out and throw isotopic enriched stuff all over the place. This is just what occurs naturally. And the theory here is that we can pick the mushroom, grind it up, measure the N15 in the mushroom. And this tells us something about where that nitrogen came from. So if the number is really high, we tend to think that's organic nitrogen. So in that first example with Douglas fir, you're on a low fertility soil, and you've got piloderma and all those things, they're the ones, their signature would be more like 10. Whereas if you're in that really rich soil, like that Grand Fir site, you're down here around 1 to 0. So that suggests that it's much more inorganic nitrogen is supplying nitrogen to the tree. And so we sampled a bunch of these types of sites on the West Coast. And for sure, this group of fungi consistently comes down very low. Some of these anosebes, Clavulina, Coralloides to some degree, Amanita constrictor is a good one, Arustula, Hygrophis. Some of these are very low. So that just suggests that, yeah, they're getting nitrogen from the inorganic sources we would expect from that other study. Whereas these ones, Cantharellus, a lot of the Cortinarius are considered, nitrogen poor species, and they're coming in around 8 or 10. They're not taking up inorganic nitrogen, they're going for that organic nitrogen. So it just shows a nice array of traits across sites. And then I had this brainstorm. N15 is really tricky and hard to interpret sometimes. I said, why don't we just measure nitrogen concentration of the mushroom, right? Because we do that with trees all the time. The more nitrogen in the tree, the happier the tree seems to be. What if more nitrogen in the mushroom, more happy than fungus is? So we sampled that same gradient across southern Vancouver Island, and we went from very low nitrogen content to very high nitrogen content. And we did find, and I tried saprotrophic fungi as well. That's kind of my control in this case, because saprotrophic fungi are living independently. They're not attached to the tree. Whereas the ectomycorrhizal species, the diamonds here, are the true symbiotes. So a couple things. One is there's way less nitrogen in an ectomycorrhizal fungus. So this is many dozens of species, by the way, much less than the saprotrophic fungi. So that makes perfect sense because the symbiotic fungi, the whole purpose of a symbiotic fungi is to take nitrogen from the soil and give it to the tree. It's a conduit to move nitrogen into the tree. Saprotrophic fungi are only living in it for themselves. They're not sharing things with anybody. So they don't need to hand off nitrogen. They keep it. So they have way more nitrogen in their mushroom than the ectomycorrhizal. So that was a nice confirmation that these are doing something important. And then secondly, it did increase a little, not as steep for the ectos as the saps, but good evidence that on a really rich soil, you're going to have more nitrogen in the mushroom than on a really poor soil. So it's a bit of a reflection of the site itself. And then we looked at phosphorus as well, but what really jumped out at me was this NP ratio again. So remember I was saying earlier, if you're a low NP, it means you're starved for nitrogen. If you're at a high NP, you're starved for phosphorus. So it's sort of a nice way of gauging that trade-off. Well, I looked at NP of the needles of the Douglas fir, and then I took the NP of the mushrooms. And again, this is a bunch of species from a plot. So that's the community average. And it's almost a one-to-one relationship. So that's a really nice, to me anyways, a nice demonstration that the symbiotic fungi and the tree host are completely as one. They're completely sharing the environment equally amongst themselves. So the tree is not just stealing resources from the fungus and leaving the fungus high and dry, but the fungus on the same token is giving what it can, is sharing that, if there's a lot of nitrogen, we'll share it. If there's not a lot of nitrogen, we'll share less. And so the stoichiometry is really quite coolly aligned. Some people have used this term holobiont. I don't know if you've ever come across that. It's not catching on as far as I know, but it just trying to capture this idea that we are talking, some people use the word supraorganism, which is probably too loaded a word, but it's, you just have to get past that idea that the tree is just an individual, like it's just a lone individual. Clearly, a tree is an assemblage of species. There's the tree, and then there's a few dozen ectomycorrhizal species on its roots, and they are all in it together. You really can't divorce one from the other. And of course, those ectomycorrhizal fungi do not grow without a tree. You never find them and you can't culture them very well. They don't grow. And in the reverse is true as well. Those trees will not survive without their symbiotic fungi. So the holobiont is this idea that it's really, really integrated. The other thing when we were playing around with that came up, I was collecting, this is, oops, sorry. This is Lactarius babicidus. Yes, this is Amanita constricta. This is Anosebe maxima, I think is the new name. It was hirsuta. This is one of these Ramaria gelanartia or something like that. And I was blasting these things and I kept noticing when you look at these distribution maps, they're always from the West Coast, hey? So we call these endemics. And I was shocked by how many things we collected in the wild were endemic to the West Coast. You sort of expect a lot of them would be circumpolar in distribution. But I'd say half the species I collected on the wet side of the island, like around Tofino, Banfield, that kind of area, say half the species were endemic, which is kind of shocking. And then in the reservoir, when we did that study locally, it was something similar, 40, 50%. Now, these databases are getting upgraded all the time and people are sending in more sequences. So sometimes what looked to be endemic later turns out not to be, was just rare. But nevertheless, it's a shocking amount of endemism, really, when you think. I don't know what my guess would have been, 5%, maybe you're endemic, but it's a lot more than that. So then we did another study. This is another student I worked with at UVic. This is published in New Phytologist. So we were working again in the wet forests on the west side of the island, looking at Sitka spruce and western hemlock. And we collected root tips and mushrooms to look at this endemic community. What are they like? What are their attributes? And we used sporocarp again. So the nitrogen concentration of the sporocarp, the endemics are in blue, Amanita, a lot of these anosebes. And the yellow are more cosmopolitan species, so they're found, either in Eastern North America or some parts of Asia or wherever, but they're not just limited here. And, the top 6 were endemic, and even like the top 9 of the top 12 were endemic. So heavily skewed to that endemic. It's not perfectly universal. Some of these, like Cantharellus, is considered an endemic, but didn't have a particularly good one. So it's not a 100% rule, but certainly a lot of that endemic community was skewed that way. And when we sampled 5 sites, and the red is the endemic of this one, the blue is the cosmopolitan. So there was a 24% increase in nitrogen on average for the endemic fungi. So clearly, we call these now the sort of specialists. They are a cut above in terms of their traits. Their ability to bring nitrogen from the soil into the tree is that much better. And this is pretty high nitrogen concentration. This is up around 6%, a lot of these. So these are really productive sites that we sampled. We did that on purpose. We wanted to look at rich ecosystems as kind of an example of how endemics might do. So it's actually a stronger signal than I got in that first study because we focused on those richer sites. And so the upshot is these endemic species are just well adapted to coastal soils. There's more data in that paper, but I just wanted to show the nitrogen. So that's just another idea, like when we think about what are these fungi, why is there so many fungi, what are they up to? It's another good point to make. Okay, so that was my whole part on what do these fungi do. The other part of my talk, how do we usually take a little break or we're good? Okay, I got about another, I don't know, 10 or 15 minutes. Is my other obsession is just biodiversity and the conservation of these fungi, right? Simply because they exist and they deserve to exist. And who are we to extirpate them or, God forbid, make them go extinct? I like to think as a conscientious land manager, the Forest Service, that if we go into a valley and we say, there were 500 species of ectomycorrhiza when we showed up, and by gum, there'll be 500 when we leave, right? We don't want to just let things fall off for the simple production of timber. I think we should try and keep these things around as best we can. That's Paul Kroger. You must know Paul, right? Does he speak here? These are that's Charmin Gamete, who you might also know. These are some other people I worked with in the Kispiox. And that's Claire again from UVic. So we like looking at mushrooms. So this is retention forestry. And this has been out for a couple of decades, this idea of leaving blobs of mature forest in cutovers like this, leaving habitat behind as lifeboats. There's a nice paper by Linda Meyer where it said retention forestry, what are the most significant changes in forest practice since the onset? And I tend to agree with that. It's a nice pivot away from just cutting everything down as fast as you can. So I have worked on this a bit because it's a great theory that if you leave stuff behind, surely the mushrooms will be left behind. But it's nice to look into these things in some detail and make sure it's working. So this is the mass study up near Campbell River in the Montane stands. So it was Amabilis fir, Western hemlock, where I found that Gymphidius. And they left these retention strips. So these are old growth strips, about 160 meters long and 50 meters wide, as well as a pretty decent blob of old growth. And it looks something like this. So that's the retention strip. It's a little bit airy, you know, it's narrow enough you can kind of see the sunlight coming through, but there's still lots of big trees in there. So it's a nice lifeboat, as they say. The openings are, I keep doing that, sorry. The openings are pretty brushy, hard to walk through. They're about 30 years old now, so they're dense. Their canopy's not still kind of to the ground. And then that's something what the unharvested stand would look like. And so I tried something new with this study that I'm kind of excited about, and that is spore traps. So this is a pretty simple concept. You go to Canadian Tire and buy some nails and wire and put this filter paper out for a few weeks, and the spores just passively drop on that filter paper. Then I give it to this wizard I work with at PFC, at the Pacific Forestry Center, and he pulls the environmental DNA out of that filter paper and tells me what fungal species are nearby. And I don't have to tell you, this study could not have happened 10 years ago. It would have been impossible, right? Like these are just this environmental DNA is just wickedly cool for trying to tease out some of these questions. It's a little bit different than when you just sequence a mushroom by itself. Now we're just pulling all the DNA out and running it together. And the computing power, like the computations you have to use to take that blob of DNA and figure out what all the species are, is beyond me. I've decided I'm too old. I can't learn these things. I'm barely got a cell phone. I can't learn new stuff. So I'm definitely working with some smart people now who I'll do the grunt work. I'll go put them outside, and then I give the filter papers to them, and they tell me what they found. That's the way I should have been doing it from day one, really, when you think about it. And so for the ectomycorrhizal, this is just year one. I'm going to do a couple more years because, you know, there's a lot of variation year to year. But like this nice Ramaria salivariescens came up, and this Hygroceras pisceae. At 144 species that first year. It's way more than I was expecting. Incredible. And that was a dry fall. This was 2 years ago now. A dry fall. And I didn't see that many mushrooms as I was walking around, but there was lots. And so this is an ordination, which is just a tricky way of taking community data and comparing it. That's my stats update. So this is the opening. So where those retention strips were, I went 50 meters into the cutover and put those same traps. And we had quite a different community of ectomycorrhizal species in the opening compared to the patch, is the blue one, and the old growth is the red one. So that kind of confirms this idea that a young stand does not have the same biodiversity, the same community as an old stand, which is something we've always argued. Once you cut a tree down, you restart succession. And, you know, I fully believe that a lot of that fungi will come back, but there's always this initial massive change in the community. And so what strikes me as interesting is that these mushrooms, like this Ramaria that's fruiting in the strip, that retention strip, those spores are not getting into that opening. So a lot of these cool species of mushrooms, I think they have a very short dispersal distance. They only go a couple meters. And so that's partly why succession takes so long. Because if they just threw out, you know, your general image of a mushroom is it just creates billion spores and they float everywhere and everything is everywhere. But when you do this kind of work, you realize, no, they're not getting past that much outside that retention strip. So we'll see. I've got 2 more years on this, and I hope I don't come back in a few years and completely change my story, but at this point, that's my finding. We'll see. The other thing that I tried here is polypores, these big conch fungi. I couldn't key out a polypore to save my life. Way too hard. Plus, they're hard to find. You can imagine crawling through the stand under logs trying to find these polypores. I got 72 species of polypores in those filter papers that first year. So there is no way I would have found 72 species on my own. And not to mention key them out. You not only find them, put a name to it, right? So that to me is just shockingly effective. And in this case, the communities, there was quite a bit more overlap. Again, that's just year one, so that may change. But it kind of makes sense to me because the cutovers have a ton of dead wood. The stumps themselves have all kinds of conchs on them. So in terms of habitat, a young stand, especially after this first rotation, still has a lot of big dead wood. In Europe, they get very excited by polypores. They often have them red listed. They use them for markers of natural habitat. But in Europe, you're often looking at third or fourth rotation. So the big wood's long gone. You really are dealing with, tree farms. So that's a caveat, of course, with any kind of study like this. That's just the first rotation. If we do the study 3 rotations from now, we might find the differences just keep stronger over time. But at least we have some cool methods to try and sort that out. And then another study looking at the effects of cutting down trees. This is Sycamore Creek, which is in the interior. So this diverts from my topic heading of coastal British Columbia, but bear with me. I was working with a student on this. We used these small 1 hectare openings, and we did a transect study where you go from the forest into the opening. We did 9 openings like this. And these, again, are about 30 years old. We wanted to get a sense of how much has that community recovered from the old forest compared to the young forest after 30 years. Pretty slow growing, high elevation stands. The rotation on these is probably like 100 years. And so that's the distance. As you go into the forest, we did every 10 meters into the forest, and then this is into the opening. So you're almost at the center of that 50 meters would be the exact center. So a couple of things that were kind of cool here. One is we actually had a bump in richness. This is ectomycorrhizal richness on root tips, okay? Fungal richness. There was a bump at the middle, but you actually find that in the literature a lot with plants and birds and all kinds of things. When you have that ecotone, we have closed forest and open forest habitat smooched up together, you get this intermix of species, early sterile species and late sterile species. So it's actually pretty common to find a bump in species richness right at the interface. So we're finding that for ectomycorrhiza too, which is good. And then it drops off dramatically as soon as you get in that opening. And so it's about 15 species per pore. Here it's more like 20-ish. So it's overall, about a 30% decline in average richness at each transect. So even after 30 years, clear in a small opening, you're only 50 meters away from mature sand. A lot of those species have not bounced back yet. On the other hand, a lot of species have. So it depends if you're a half glass empty or half glass full kind of guy. I actually thought, especially given the size of those trees, I didn't think we'd find much diversity in those openings at all. So there are definitely a lot of multi-sural species, what we would call it, any species that can kind of grow anywhere, as long as there's a host. There are definitely a lot of multi-sural species in our ecosystems. And this again is a community ordination diagram. So the opening, and then that's the edge, and then that's the forest. So there's this nice gradation, just showing the successional pattern again as you go from the forest into the opening. And a species like the gypsy mushroom, there was about 10 or so species that are common enough in the stand that you can see on the root tips how they change. So they're gypsy mushroom, we have found lots in the forest, handful in the edge, and then nothing in the opening. So it's one of these species that's clearly sensitive to logging. So those can be nice indicators. Of course, there's also a lot of indicators that aren't common, like Romeria, and they're virtually impossible to track on root tips. So you have to go look for them to decide. You have to walk all those openings and say, I found 50 ramaria in the forest. I didn't find a single one in the opening, and we can only do that through spore park surveys. But this at least shows that this succession is going on. And then this was a chronosequence study. And this is generally how we do this kind of work, like just find stands of different ages, hopefully with similar soils. They're never exactly the same, so that always adds a lot of noise. But find different ages and look at the community and try and sort, what's the effect of cutting and how long does it take to come back. And this one we did quite a while ago. This was sort of before we had all these molecular tools. So we were basically putting names to species and doing our best. So if I was to do the study today, I'd have a lot better result, I'm sure. But I think the general pattern would hold. And that is that, mushroom richness increases pretty fast. So those early years, a lot of things are roaring back, as I was showing in that signal. And then somewhere around 75 years to 100 years, you tend to plateau, and the community gets pretty stable. So it's kind of good news in one way, in that it's not like if you go to a 500-year-old stand, you find even more differences than a 100-year-old stand, as best as we know. And I have to admit, we could do more research on that, but as best as we can tell, it tends to stabilize. So really, you just need that early part of, when we talk about old growth, I often say, well, I would say mature at least. Mature stands, you get a lot of diversity. And so, these young stands, tons of species, and then we just kept accumulating species as we went. And I would say, like in this case, 125, 220, these were the same communities. And kind of gets back to the idea that you need canopy closure, so you need interior forest habitat. They seem to need the shade, the moisture, the mosses, that habitat for the species like the gypsy mushroom seems to be pretty important. And then once you have that habitat, then you take time. So in that Campbell River study, I was showing that the spore dispersal seems to be very discrete. And so you have to think, say, somewhere around 50 years, okay, we have the habitat, we can all come back. And so they slowly start walking back into that stand. So it takes some time for those things. And the other part of this too is, like we found, for example, a pine mushroom. You can sometimes find a pine mushroom in a 50-year-old stand, let's say, but maybe just one. Whereas you go into these older stands and there's sheros all over the place. So there's still a distinction maybe between, yes, it's back, versus yes, it's at its inherent abundance. That again takes more time. So we have to be careful when we talk about just richness, as if, as long as they're there, it's all good. It's nice that they're there and they're back to the abundance they were sort of pre-disturbance, if you're really talking about recovery. So my guess is, and again, this is a bit of a guess, I'd say maybe 15% of the community, I would say, is in this oldest age class, like sort of 75, maybe 100 plus. So it's not a ton of species, I think. And a lot of it, albatrellus, some certain amanitas, boletopsis, the ramaria is pretty good. Some of the tricholomas, there's basically species in a lot of different genera that really seem to be late Searle. Not late Searle like 500 years old, but late Searle 100 plus, I would say. So it's not a ton of species, but they're really nice species. They're often endemic species. They're uncommon. They're nice to look at. So that to me is what we're trying to protect with retention forestry and some of these other tools. Okay. Some conclusions. I don't think I've ever talked this long in my life. Everyone's still with me, so that's great. Thank you for that. Some conclusions. So I'm convinced, and I hope you are, that these communities are not just Mother Nature being superfluous. It is highly adapted to the full range of soil nutrient availability. These inorganic nutrients and organic nutrients. And both when they're in high surplus and it's a very competitive environment, and then when they're scarce and it's a very difficult environment. Really important conclusion. And as it turns out, considerable number of these special species are endemic to the West Coast. So if we lose them, they're irreplaceable. I don't know if you could truly lose them, but certainly if you did massive clear cuts with no retention and short rotations, the sort of old style logging. If you kept that up for a while, I'm sure you'd start losing some of those specialist species. We don't do that anymore, I would say, but nevertheless, these are the species we need to keep an eye on. Make sure that they're bouncing back the way they should be. And the functional ecology, so tree species are ectomycorrhiza, but we do have some arbuscular, like western red cedar, yellow cedar, big leaf maple, another arbuscular one. Very distinct ecology. So it's kind of fascinating to think about how do they even coexist, right? They're completely different traits. Yet we see them growing together all the time. And maybe they kind of feed off each other in some ways. But definitely from the work I've done, it's night and day in what they can do. I pretty strong support of retention forestry and the conservation of those late, especially those late seral species. Those are the ones we need to work with. And in that sycamore study where we even had small openings and it was taking its sweet time, I do think, we should just admit that these ecosystems probably take close to a full century to fully bounce back to that sort of pre-disturbance condition. So as you probably would know, rotation age, the way we like to do forestry, is never a century. So that's kind of a challenge. And it's an economic challenge, but I always talk about ecological rotation. Don't just think about when does the best time to cut down the tree to make the most money making plywood. If you were being compassionate about it, you'd say, when's the best time to cut out a tree? When the community has had a chance to recover, kind of come back to what it was. So sort of working with nature rather than just imposing mankind's needs. That's, I don't know if that will ever happen, but ecological rotation makes sense. And I think that was it. Oh, one more thing. This is a nice picture at Thetis Lake. And when I take students out, sometimes when I'm walking around, I like to point this out. This is really terrible soil. It's rocky, coarse textured, not that much organic matter, very low nitrogen availability. And you have these monster Douglas fir on here, right? If you were a farmer and you logged that off and built a little cabin, had a couple of goats and planted some potatoes, you would starve to death. That is terrible soil, right? And so I just think, to me, this is 50 million years, give or take, 50 million years of evolution. Mother Nature has been trying to sort this out for a long time. How do we maximize life on these lousy soils? And she's done it through ectomycorrhizal fungi. And I will give some credit to Douglas fir, but it's that symbiosis that is just tremendous. Like the amount of carbon fixed on a hectare of lousy soil like that is mind-boggling when you think of what mankind does with goats and all the rest of it. So I just think it's a marvel that we don't often stop. Okay.
Thank you.
Ooh.
You just see
That's right.
We're gonna give you a mic so others online can hear you. So
I think that mic just died.
it did just die on us. Apologies.
Did you discuss sealed stumps?
Sealed stumps? Like the knobby stumps?
Yes, it
Oh.
does. Yeah.
It did. That's okay.
Well,
We can stop
I'm
the
not an
recording.
expert on that, but my understanding is it's root grafting where the roots of the trees will bind together. I see that with Douglas fir more than any other species. I don't know if you know that, Andy, but I like with hemlock.
Fungus would be involved because it seems to be restricted to this.
Let's just ask some questions and you can
Right.
use the computer itself.
I
You can-
think mostly it's root grafting, like the roots actually.
But it is
Right,
curious
but
as
we-
to,
because it doesn't seem to happen anywhere else.
yeah,
That's called, Andy calls that the tree's knees, I believe.
yeah.
Yeah, I think root grafting is the explanation. As opposed to, fungal connections.
Right.
But then we don't know why it only happens here.
Yeah, it's a good question, really. Yes. When you talked about the energy, flow of energy, you implied that there was a lot more energy from the tree to provide to the microbial network. Is there some mechanism there to utilize for carbon simulation? Like, and that if you could facilitate the health of the mycorrhizal on the tree, could you assume more carbon?
Carbon into the soil.
Yeah.
Yeah, I would say that's a pretty active area of research, trying to sort out how much organic matter in the soil comes from the tree and the fungus. The root tips themselves die off and replace themselves quite readily, like every year or so. So there's a huge amount of root litter. And a lot of that carbon was from the tree, basically. So the root litter is an expression of that carbon. And then there is also people who argue that the roots are kind of leaky in a sense, that especially late in the summer when the tree stops growing, it's still photosynthesizing, I guess. And so it will sort of let go of some of that photosynthetic carbon, and it'll just kind of go down to the roots and just go into the soil. It's kind of fascinating. It's also very difficult to measure. So I don't know how you do a good job as a study, but it makes perfect sense to me. That, yeah, it is a carbon pump. It's just put stuff in.
Would you be willing to hazard a guess at like how, like, is it, you know what I mean, is it really important from your point of view, despite it being hard to measure?
I would say it's really important. Yeah, I think a lot of that carbon breaks down pretty rapidly. I don't think it sticks around, but it's stimulating life. So all the bacteria, fungi, all the things that eat that, like the mites and calendula worms, you know. So it's basically the building blocks of the food chain in the soil. So yeah, even if it doesn't materially change your carbon concentration in the soil over time, it's still making that.
Is anybody looking at the effect of the water cycle vis-a-vis climate change? So we have the 30 by 30 people that want to have focus on 30% of the environment protected by 2030. You know. Then we have the 50-50 people saying we should do half for nature and half for humankind. So with respect to the coastal forest and the water cycle, if we have more water in the air and less in the soils, that's going to affect the NP site ratio,
Yeah.
I would suspect.
Yeah, making soils more droughty is definitely a huge concern. And I don't know what the forecast, like the models suggest. I think that, yeah, we'll get droughtier summers. They're already droughtier. So if you kind of hit a point where even the fungi give up, like this is too dry for us. But I don't know what's going to happen.
Marty, Tom Rankin claims that N15 is just a marine source
Resource.
with salmon
Right.
coming. So if your mushrooms which accumulate N15 are some which grow close to the streams and so on.
Yeah, I've read some of that. So that's a little bit of a special application because what he's arguing is that salmon have a particularly enriched N15 in the carcass. So if you look at N15 in the tree, you might see spikes where huge die off the salmon and all that nitrogen in the soil. But everywhere you go, there's N15. So the mushrooms I was showing you were probably a tiny, you know, like the concentration was still very low compared to, say, that salmon. So there's always N15 around. It's just a question of interpretation, which is part of why I stopped N15, because it's just like, well, you can almost interpret it any way you want. It's a hard thing it's used a ton. Everybody publishes N15 things. We can go in circles trying to. But anyways, the marine salmon derived nitrogen, now that's a kind of a separate use of that isotope, but I don't think Well, if I sampled mushrooms on a stream bank, maybe I would see the same spike in a mushroom. Like maybe that N15 would be, I don't know which direction, maybe it'd be like 30 ppm minus 20 ppm, depending on what the salmon. But you could, in theory, if you had enough salmon rotting in the soil, it'd show up in that mushroom for sure.
I have a lot of questions online.
Oh, yeah?
May I shoot away?
Okay.
Yeah, and we'll give everyone else a chance again. Do you believe brassicas are also mycorrhizal? My understanding was that they weren't, but research might be needed.
I think they're correct. They're considered non-mycorrhizal. It's kind of a lot of the, certainly some of the weeds that are the precursors to our agricultural plants they're non-mycorrhizal and grow just in, disturbed soil really rapidly. So yeah, there's kind of a special subgroup for sure.
Okay. What mycorrhizae are good to add to a native blueberry bush planting in the garden?
Well, now that's another type of mycorrhizae, the ericoid mycorrhizae.
Yeah.
I suspect you could buy some inoculum, I bet, for blueberries.
Tons of mycorrhizal soils. And
Yeah.
I would assume they do their due diligence.
Yeah, that's one I've never worked on, is the ericoid mycorrhizae.
Question: 1 out of 10 biomass just in our area or similar in other world regions? Not sure to where that applies.
Biomass, like the tree biomass or fungal biomass?
Either way. It says it just 1 out of 10 ratio.
At some point about a tenth of the biomass and like the ectomycorrhizal fungi and then the other part you had it's like a blue rectangle.
Right.
Yeah.
Yeah, I don't have the numbers in front of me, but some studies, I don't remember the exact ratio, but it was pretty impressive. Maybe 3 to 1 fungal to saprotrope or something like that. I probably said 10 to 1 in a dramatic moment, just trying to expound upon my point. You probably shouldn't quote me on that 10 to 1 ratio. It's definitely, I think we can say pretty confident there's always way more ectomycorrhizal biomass than saprotrope biomass in forest.
Thank you. Are the ectomycorrhizal fungi symbiotic with each other and in addition to the host tree?
I don't think so. There are occasional species that we might call parasites. So they tend to grow on the root tips and pull stuff from that. That again, people say, oh, I think that's a parasite. Then you look at their evidence, you think, I don't know if I believe you or not. So it's kind of up in the air to me.
Which brings me to the Comphidius, because you mentioned you found 2 new species. Did you ever notice any sealers nearby or anything like that?
Well, that's the thing, right? Comphidius is, it's usually on Sewillis, but there was hemlock and Abies, which doesn't generally have Sewillis. I think I'm right with that. Yeah.
It's interesting.
Which I always think of Sewillis as
is it?
pine,
Lachii?
fir, and fir. But yeah, you're right. Comphidius and Cumgalpus, I think, are considered parasites.
There's a relationship that may or may not exist because we don't know.
Yeah.
Let me see. We have the temperature impact on nitrogen composition. Temperature. I would assume temperature, summer versus fall temperature, soil temperature.
Yeah, sorry, I don't understand that.
We have a lot more than we can. Juliet mentions we can't set regional or global logging policies based on what we believe or want to believe. Which might happen either. Very true. Sub-B2 polypores is very amazing, by the way. There's just lots of comments on here.
Yeah.
All right, let me see. We got, lots of thank yous, What percentage of cut block prescriptions on Vancouver Island presently and or last 5 years using retention logging practices?
Well, a lot of public land is using what they call clear cut with reserves, which is kind of a form of retention. It's generally bigger blobs of retention than scattered. Legislation right now is 7%. You have to leave 7% of your cut blocks standing.
Fair enough.
Minimum.
It looks like there's a lot of comments that say that they've observed clear cuts. So it's one of those cut block versus clear cut situations, I'm sure.
Someone mentioned that they're concerned that Mosaic is not following new logging practices. They're near Port Alberni. Short retention, clear cutting, etc. are normal practices and happening right now, and they need new legislation for private landowners.
Yeah.
Different things to try and control.
Maybe that could be a different
Whole nother talk. Reform. No. That's about it for the online. Anyone else brewing on a question? Well, you've got Marty here.
Yeah, I've got a question, but maybe you hit on it when you were talking earlier. I noticed when collecting for our mushroom show over the years, by going back to the same places, there are what I would call hot spots. Incredible diversity of fruiting bodies in one spot, year after year after year.
Yeah.
And you must have come across.
Yeah, I
it
do that
yourself.
too. Yeah, there's a few places I like to go. Yeah.
Yeah, and you see it, and it's uncanny. At first, you don't realize it, but after the years, you go, "Well, I'll just come here for the show because it saves me a lot of time."
Yeah.
And I think that's mostly just soil, good soil.
That's a good question.
Or moisture.
Can we rule out wood nymphs? I don't know. Gotta wonder, like a tiny gap in the canopy that just gets a nice little bit of rain. You know, it's just a nice aspect. And,
There's something special about those.
yeah, I know what you mean though. There's some certain spots I just think I love going through. Lots to see.
Jay, could you say something about your traps in more detail? What influences just the layout? Because a lot of your conclusions are based on what is put in the trap.
Right.
So what influences that trap? What exactly are you putting in it to catch all this?
Well, the
Would it
filter
vary
paper
by
you
species?
just use a buffer solution first, just to try and keep the DNA from degrading. And then in the retention strip, I placed them systematically. So I had sort of one here and then 10 meters on either side. So I had 3.
How big are they?
Oh,
Each
the
one?
filter paper is 9
Oh,
centimeters.
yes.
Then I have those 3. And then I had, in each strip, I had 3 reps. So basically 9 traps in a retention strip. And then I did the same thing in the cut over next to it. And then same design in that old book. So I had 81 traps in total.
And how often do you change them? The filter?
Well, I've just been doing like a long 3 to 5 weeks in the fall. I tried to time it to sort of peak mushroom season. Campbell River, it's a little bit out of the way for me, and I have trouble going up field work. So ideally, I think if it was closer to town, I'd probably do like put them up for 2 weeks, bring them back in, put another set out for 2 weeks, and another set for 2 weeks, because that way the paper doesn't start to disintegrate. When you get those wicked storms come through, sometimes they get pretty beat up. And then you'd have like a 6-week window, and you could just collate all the information.
Because running up the rain,
Yeah.
they're very absorbent and they always fall to bits.
Yeah, I've been playing around with various things. I think this fall I'm going to just do 3 weeks no matter what. It was part of it was I was keeping my eye on the weather, and I just thought, well, it's been really dry. I don't think anything's fruiting. I'm just giving another week, and then it just rained. So this week, 3 weeks. This year, 3 weeks, and a little later because I kind of thought things started fruiting really high about mid-September, but now I think it's more like towards the end of September. So it's just one of those research sites that's so far enough away that I can't get out as much. But anyways, we'll try that 3 weeks and see what happens this year. Yeah.
On those same study sites, have you considered doing management on soil samples?
I have thought about it, but I felt like the spore deposition was a really nice part 1 kind of thing. Like what fungi are fruiting and how widely are they distributing from the fruiting and how do these habitats compare? And in my experience, when you start layering in other sources of data, it becomes so unwieldy that nobody wants to read it. So I found if the spore carp data was cool and made a lot of sense, then first we could do a second study where they sampled the roots themselves and all that. So this was sort of part 1.
We have a question far back, patiently waiting.
A bit of a strange question, but I was wondering, how do people find out if new species are edible?
Yeah.
We'll
That's a
give them
good
to you to try out.
question.
No.
Somebody has to be brave, I guess. So just start with a small amount probably and see how that goes.
And nowadays, though, we have a lot of, what would you say, history of what type of genuses are normally and species are normally considered edible. And you can sometimes work your way into that genus and be like, is this very risky or not? And then sometimes with some studies where you look at the toxins that are within, if you can afford to do so, or if people can look into it, then they might have a better idea. Ooh, oh, there's something such and such in there. Let's stay away, kind of thing.
Metabolemics is coming in more and more frequently with the new equipment that they've got. So you can get a profile of what is in any mushroom now, essentially. And that's the new equipment that's coming on, the sequencing equipment.
Does that help
And-
answer your question though, man? Yeah, okay.
And that would give you a very good idea of, like, for instance, what's on that table.
Yeah.
All right, and then you know exactly what they are, and then you can compare it. You know, you're beginning to resolve what are the toxins that are giving you that syndrome.
But could you tell if it tasted good?
No, no.
That
Taste
you-
this.
So you just
Yeah.
rule out of a killer.
Well, we know that they're, and that's the way we're trying to find it.
That's interesting.
A good example,
Oh, sorry, go ahead.
say a good example of what he's talking about is that mushroom on the table, the Geometra esculenta, which is poisonous.
Right.
But we always used to tell people, stay away from all the geometras, right? They're all poisonous, but you begin to look at the chemicals inside of them, the toxins, they're not there in most. So researchers understand that's one where it is, most others don't have it.
So I got,
Yeah.
okay.
Marty,
Go first.
one of the dominant mycorrhizal species in Ponderosa pine is Wilcoxina, which
Yeah.
is an asp.
Yeah.
Did you get any asps in here?
In really young disturbed soil, we get a lot of it. One of the first to bounce back. And in greenhouses, if you grow stuff in greenhouses, so I would call it a ruderal being.
On the roots.
Yeah, I would call it a ruderal species, but it is interesting when you get to those really dry interior forests, that ruderal species never seems to go away, which speaks to me how challenging that environment is for a lot of fungi. It's just so hot and dry that I think a lot of what we think of normal fungi can't hack it. So Tim Philpott and I are actually doing a little drought study where we're going to these really, really dry habitats just to get a sense of what even fungi persist and grow in these old forests. So I expect to find a lot of them. And you had one.
Yes, in the newer logging method where it's mandated to leave 7%.
Yeah.
Is there any specification as to where the 7% should be? Is it should it be downstream of the logged portion of the water?
I don't think there's any recommendations on where it goes.
Uh-huh.
There is always a concern that it's the rocky ridge that doesn't barely grows a tree anyways.
Right.
And that's what they'll put aside. But I think for the most part, over the landscape, you get a pretty decent cross section of habitat.
Does that 7% still include what's supposed to be left around creeks and runoffs and that sort of thing, or no? And caves and
Yeah.
dens and that sort of
I think it's separate, but maybe don't quote me on that. I think there's all kinds of provisions, sensitive areas and stuff that have their own legislation, and then the 7% is
That would make sense.
more related on.
I have another question on here.
You don't think so?
Thank you for that.
Oh, sorry.
No,
Andy said I'm wrong. Okay.
Aw.
Stop that.
that's sad. Someone else asks, how do mycorrhizae change over time with the growth and regrowth of the forest? You said that they expand slowly into the clear-cut areas or the open areas. Does the radius expand in relation to the oldest host trees, or how does that work?
Well, I think a lot of species do spread vegetatively.
Mhm.
So it's basically hyphal contact, and they'll hit new roots and colonize new roots. So I think there is some of that sort of spreading through the soil. And then there's also all the spores trying to do their thing. So it's a bit of a mishmash.
As long as the ecosystem is,
Yeah.
receptive.
And I would also say that that's almost space age kind of detail compared to what we generally try and do with our ecology study. So not that it's it's not a bad question. It's just
No, it's interesting.
there's something. A lot of questions.
Just one more, please. That 72 species of polypore, when you went into the area, could you find 72 different species?
I doubt it.
So, yeah, but where would they come from?
Oh, I think a lot of them are very
You can't see.
inconspicuous. Yeah, they're very inconspicuous.
Yes.
And they might be up the tree, they might be under the underside of a log, they might be.
I just wondered
Tiny,
if
I
there was
don't
any correlation with what you're finding or whether it's coming from somewhere else.
Well, if it was coming from somewhere else, then you would sort of expect to see it on all the traps. Yeah. So that'll definitely be a reasonable conclusion. You just find the same species everywhere. Then you would sort of think maybe these spores just are like dust. But at this point, I would say the ectomycorrhizal community seems to be more specific to habitat than polypores. We'll see how it goes. But that's the beauty of this kind of spore deposition studies, that I don't have the time or the expertise to go find 72 polypores. Just nobody does, right? It's extraordinarily challenging in that environment to do a proper survey. So just to look at the spores is kind of silly. I don't think it's perfect. We probably miss stuff, but still, hopefully it gets you to your answer more readily.
You should get some of us from the club.
Yeah.
to help you.
Yeah, we do have a conservation committee and people would be happy to volunteer.
All right.
Yeah, it's great work.
Forever. Who's with me?
Yeah, you'd be surprised. You'd be surprised. Yeah.
Tomorrow.
Tomorrow, yeah.
What's the process for, you mentioned all these undescribed species. What's the process to just kind of
Well, there are a lot of taxonomists out there in the world, and I think I've had emails from time to time, people just say, can you tell me more about this mushroom you found? But for the most part, I just put it out. I put it at UBC, so it's readily accessible for any taxonomist. And then my photo and my sequence goes in the scope of the sense. And hopefully someone someday will put a name to it. I don't know who else to probe, prod.
And you don't feel you can name it?
No, I'm not a taxonomist. That's a whole different. It's just the lack of knowledge. Yeah, and I think if you're doing it properly, you would sort of want to check all the other herbariums to see if somebody's already named it and put it somewhere and didn't sequence it. It would be a lot.
Yeah. That's a wrap online.
All right.
That's it. Thank you so much.
Thank you.
Half expecting people to start leaving.
No.
It's taking too long.
Just close the door.